Okay, really terrific. And as we continue the theme here, now we're going to switch to a panel discussion, point of care monitoring and continuous monitoring. Our session chair is going to be Vic Bajaj. Vic is Managing Director of Foresite, who evaluates and pursues investments at the intersection of technology and life sciences, including in personalized and precision healthcare. Before joining Foresite, Vic was Chief Scientific Officer at Grail, a life sciences company a lot of us have heard about, to detect cancer early when it can be cured. He holds appointments here at the Stanford School of Medicine, as well as Lawrence Berkeley National Lab, and he got his PhD in physical chemistry at MIT. Welcome, Vic.
So good to see all of you, a lot of you for the first time, but also some familiar faces in the audience. So we're going to talk about today with a distinguished panel is the idea of point of care analysis and precision continuous monitoring. And it's interesting, I think we have a unique perspective at Stanford and also in Silicon Valley on this topic because we are often motivated by science fiction ideas of what healthcare can be. The idea that we can measure early biomarkers of disease and quantify that and understand disease trajectories, maybe even intercepting disease early before all of the harms are manifest, that motivates a lot of this work. The idea that at the point of care, we should be able to measure analytes that are very specific about disease diagnoses. That's also very interesting. And quantify both disease severity and also suggest treatments in ways that are impossible by symptomatic presentation alone or evaluation of symptoms. That's motivated a lot of the work, but then what's the reality? The reality of medicine today is compared to that vision, it's very data poor, certainly about the individual. There are even screening technologies that do not need that frequency of measurement, things like annual cancer screening or screening for dyslipidemias, where there are very easy, straightforward interventions that don't reach a large enough population. And then where we have point of care analysis, it's mostly in infectious disease, where we have continuous monitoring of things. It is either in the fitness space, in the consumer space, or with limited applications in medically validated contexts, things like continuous glucose monitoring, you'll hear about that, AFib, but for the most part, it's things like this device I'm wearing, which for all of its continuous measurements just tells me basically that I have to sleep more and exercise more. So it's not very useful at the moment. So that's the question that we'll discuss today. What is the state of the art? What is possible on the horizon? And more importantly, given the nature of our panel, what are like actual practical business models to translate some of the phenomenal technologies that you're developing here into real products that benefit people at scale? And we'll take your questions throughout that. So the first panelist is Carrie Egan, CEO of Teal Health.
Thank you for having me here. It's been a while. Well, I guess I went to Joe's class earlier this year, but I went to Stanford undergrad 20 years ago, so it's nice to be back on campus, though it changes quite frequently. So I'm Kara. I'm the CEO of Teal Health, and what we're doing is we're redesigning women's health, starting with cervical cancer screenings. Next slide. How do I do it? Oh, perfect. So for us, we don't think women's health is a wellness benefit. We believe it's a critical piece of preventive care. And what we've seen to date is almost every women's health company is nipping at the edges and going after women who can pay out of pocket, when really there's a very massive problem that we need to be addressing, which is getting women the access and health that they need. However, the next part is there's actually a code for this. So back to business models that we'll talk about later. And it's the Well Woman Exam. And it's 100% covered for all women annually as part of the Affordable Care Act. But in the US, 1/2 of all counties doesn't have an OB-GYN to deliver a baby, let alone provide women-specific care. So we're making it accessible via telehealth. Cervical cancer screening is the primary entry point for this annual exam, and there are 92 million women who are of the screening age, 25 to 65 years old. However, they hate the current experience. For those of you who don't know, this is a speculum. It is not an archaic well, it is an archaic device, but this is actually the one that we use. Occasionally it is plastic, but for the most part it is metal, and it looks like this, and it is wildly uncomfortable for most women. It hasn't been updated since the 1940s, and it's failing women. So 75% of women have more than one barrier for why they don't want to get a cervical cancer screening or an annual visit. 1 in 3 were not up to date on their cervical cancer screening pre-pandemic. And post-pandemic, we did a 94% decline in cervical cancer screenings. So we have a current standard of care that is currently not working. We have a healthcare system that is underwater because of COVID, and we have a massive backlog that simply can't be met with the current situation. So we believe it's time to create a women's health platform that puts a woman and her experience at the center. So what we've done is we've created a thoughtfully designed at-home cervical cancer screening that will increase adherence and engagement. We've tested in over 100 women, and so far 87% said they'd be more likely to be screened if Teal was an option. 100% said it was easy or very easy to use, and we had 100% sample adequacy for both HPV and pap smear. This is how it works. So you'll either find Teal on your own by ordering a kit online, or we work with a lot of we will work with a lot of providers. So we'll talk about business model a little bit later on, but for us, the providers are very happy to refer this out. It's sort of similar to a Cologuard experience. So then we send the kit at home. One of the things that we think about with technology and what technology can do to help is really creating these closed-loop systems to understand whether or not somebody's taking an action. So we'll do personal outreach. And so for a woman, it might be like, hi, Tanya, it's 8 p.m., so instead of reaching you at 11 when you're working one of your 2 jobs, we'll say it's 8 p.m., I'm sure it's been a long day, but we saw that the kit got there. Do you have 5 minutes to go into the bathroom? We're standing by if you have any questions. That's all fully automated, but we will support people who are a little more cost effective. So highly scalable, but also highly personal. Then you collect your own sample. 90% of women can do their sample collection in under 2 minutes and put it into the mail. We then become your provider. So we prescribe the test and the kit, and then we also talk to you about your results. So we make sure that you understand if you do have a positive result, what that means and what your next steps are. Similar to step 2, on step 5, we also make sure that you've actually taken those steps. So half of all women who have a follow-up after a positive cervical cancer screening don't take the follow-up step within the next 12 months. And so for us, we're your doctor. We know if we've gotten your results back. We know, depending on what our integration is with the other health system, whether or not you've scheduled. So we can continue to reach out to you. We can make it easier for you to schedule for patient navigation. Or sometimes it's just about creating the content to make sure someone understands how important this is. So there was a study in 2016, which is crazy that in 2016 they were sending DVDs to people's houses to explain the importance of a mammogram. But the women who watched watched the video were 6x more likely to get a mammogram. So for us, we think about how do we send tailored content that really addresses women's concerns on their emotional journey to getting care and making sure that they understand the steps and the importance of it. This is kind of the beauty of what we're doing. No one else can actually unlock this Well Woman. The actual Well Woman exam has these components on I do not know my rights and left, this side, the left for you, I guess, right? I don't know. So all of it actually can be done with the exception of cervical cancer screening via telehealth. And so we're able to unlock this code that has no cost sharing to any woman, offer her a much better experience than the current standard of care, and increase access to her getting more informed information about her health. A big piece of this too around the breast cancer and colon cancer screening is in some states you have to have an annual visit before you can get those things referred. So if you think about the barriers to accessing care, about being able to attend a doctor's appointment, we've reduced that barrier. So in addition to giving you that little video about the importance of mammograms, we've also said basically if you can only handle one appointment because of your work schedule, make sure it's a mammogram and go get that done. We are currently teeing up our pivotal study for our FDA approval. And so that is a pretty straightforward study of which we've already been getting very positive results through our pilot. So we anticipate hopefully being through the FDA by the end of next year. Or end of this year, sorry. No, sorry, end of next year. The goal is that we go over time with you. And so the annual Well Woman allows us to hit you with your cervical cancer screenings, but also address your needs as a woman as you go. So that could be fertility screening, it could be pre and postpartum, it could be mental health, perimenopause, menopause. So just really making sure that women understand what's available to them. And sort of how we were talking about, or we will talk about, is the idea that we want to be this central platform to help innovation come out. So it's not that we're going to create everything, but we're going to use this platform where we can actually bring millions of women on, have them be engaged in their health, and then use data to see what would be a good solution for them. Some of it might be stuff that we would handle, like discussing fertility, but for other people, it might be, is there an early cancer screening based on your family history that's now available? We can help surface that up to you. Or when I think about GRAIL, mammograms are universally disliked, but there's a lot of money in the healthcare system in doing them. And so as soon as there's a better opportunity for a woman and a woman's experience, we will make sure that she's offered that as a solution. So we can really kind of use both data to inform who's eligible for what offering, and then also proactively reach out to them in a high-touch way with the text and automation. We play really well in the health ecosystem. So interestingly, and again, we'll talk about this a little bit, I think, later on around business models, but PCPs, typically this woman's health, because there aren't enough OB-GYNs, is being done by a PCP. A PCP does this as part of their annual visit. They actually make no additional money by doing a cervical cancer screening. So they're all more than happy to send it over to us. And then we think that there's room for telehealth, but we also think it's really important that there's times where you're seeing your doctor in person. We just don't think the cervical cancer screening is one of them. And so we play really nicely in making sure that we're referring our patients back out to the care they need in person within the existing health system. This is the device, just a little bit more about it. So we actually, it initially came out of Stanford Biodesign, and then we redesigned it with IDEO. And made it very easy for a woman to use it. So 100% said very easy, 100% said somewhat or very comfortable. 100% took less than 5 minutes. The sponge allows us to collect a lot of cells. This allows us to do a pap smear, which is a triage step, or Syntec, which is Roche's new cytology offering. And it also allows us to bring it at home because we're able to reconstitute the cells off the sample. And then we also have this mini speculum design that allows us to target cervical cells. So while we'll first go through the FDA and primary HPV, which is the leading cancer screening guideline, we also can take the triage step of pap smear over time as well. I think that's it.
So our next speaker is Marc Taub, who's Vice President of the Abbott Diabetes Care franchise.
Hello, everybody. I'm Marc Taub. I head up technical operations at Abbott Diabetes Care. That means I'm responsible for R&D, medical affairs, and clinical affairs within Abbott's Diabetes Care division. I want to thank the organizers for bringing me here today. I also have a Stanford connection. I spent 5 years across Campus Drive in the Department of Material Science with Professor Rainer Dauskart. Got my PhD there. I like to tell people that I'm on my first job. After finishing grad school, I went to TheraSense, which was acquired by Abbott. So I really am on my first job. And I've spent my entire career helping people with diabetes avoid the dreaded finger stick. We all know that people with diabetes, that it's very important to monitor and understand your glucose values so that you can understand what food and exercise and medication is doing for how it's affecting your glucose levels. For a long time, the holy grail of diabetes has been the closed loop, linking a sensor, a glucose sensor, to insulin delivery to drive essentially personalized care of delivering appropriate levels of insulin in response to the glucose needs of the patient. This is a famous photo that you've probably all seen before from 1963 from Arnold Kadish with intravenous insulin delivered and glucose assessed, analyzed extracorporeally. Here's where we are today. This is a patient, this is an ad for a patient wearing a Freestyle Libre 3 sensor, a tiny glucose sensor on the back of the arm that measures glucose readings once a minute. The glucose values are sent to their cell phone. The cell phone contains an algorithm that determines the appropriate amount of insulin to deliver based upon the glucose reading, their history of insulin delivered, and their meal. It's then delivered via the small insulin pump that's on this patient's hip. This product is available in Europe today, the Freestyle Libre 3 sensor, driving it with an app developed by Roman Havork at Cambridge University called MyLife Cam APS FX, and the insulin is delivered with an Ipsomed pump. Of course, it's been a lot of work to get to this stage, and some of that work was done right here at Stanford. This is one of my favorite photos from Dr. Bruce Buckingham. So this was, I think, probably from 2008. There's a young study subject wearing 2 devices. Those are Freestyle Navigator devices. They're essentially the predecessor of the Freestyle Libre device. You can see they're a lot larger than the Freestyle Libre 3 device. They're probably about a dozen times as large. And those sensors are talking to that receiver on the back, essentially in that white cradle on the desk. And then that white cradle is essentially a big serial cable that's getting the data into the laptop. The laptop had the closed loop algorithm living on it. And at that point in time, there was a healthcare provider, physician, seeing every insulin dosing recommendation made by the pump and hitting OK, allowing it to happen. So we've come a long way since those days. In fact, here's a system that was just approved by the FDA last month, the Beta Bionics system that uses their pump and their app, along with an ICGM approved, an FDA approved ICGM device to deliver insulin matching the patient's needs. It's approved for type 1 diabetes for children as young as 6 years of age. There are currently 4 companies with legally marketed continuous glucose monitoring systems in the US. Abbott, the company that I'm here from, Dexcom, Medtronic, and Senseonics. The devices from Abbott, Dexcom, Medtronic are all transcutaneous sensors that are applied by the patient and removed once their wear life is over. For Abbott's system, it's 14 days. We recently got approved for a 15th day. For Dexcom, it's a 10-day device, and Medtronic, a 7-day device. The Senseonics device differs in that it's an implantable sensor implanted by the healthcare team under the skin for 180 days, and then there's a transmitter that's applied over the device that powers it and essentially has Bluetooth communication to the phone to allow the glucose readings to be seen. The very first continuous glucose monitoring system was the MiniMed CGMS, continuous glucose monitoring system. It was approved by the FDA in 1999. It was a retrospective device that was applied in the physician's office. There was a small sensor that was worn on the abdomen, and then the patient wore this device, a data logger, essentially like a pager, if you remember what pagers were, on their belt clip. And then what they did was the patient went home, they worked for 3 days, and they continued their finger sticks. They needed to test, I believe it was 4 times a day. And then when they came back after 3 days, the system automatically used those finger sticks, those 4 times 3 finger sticks, to calibrate the sensor, to retrospectively calibrate the sensor to provide accurate glucose readings over what the sensor had seen, the glucose readings the person had experienced over those preceding 3 days. This is a picture of the Freestyle Libre system. This system was introduced in Europe in 2014, cleared in the US initially in 2017. And I think it's fair to say that this really brought in the boom in continuous glucose monitoring that we see today. It was very nice to hear the keynote presentation where there was a lot of talk about access and affordability. This system really was designed with access and affordability in mind. It's part of the reason that it needed to be 14 days, a big engineering challenge for my team. It was one of the reasons it needed to be factory calibrated. So all other continuous glucose monitoring systems before that time needed finger sticks to calibrate the sensor. Well, if you're trying to offer a sensor at a price that's kind of roughly equivalent to finger sticks, if you have to add in the cost of finger sticks, that makes it incredibly difficult to do. So factory calibration, 14 days were all critical in designing the device for access and affordability. And since then, we've seen really a sea change in the use of continuous glucose monitoring as the standard of care, and products like the Freestyle Libre have really changed that, where it's not just a niche product for a very small subset of type 1 diabetes, but it's really becoming the standard of care for all diabetes. Okay, and there's been a long series of innovations that we've introduced subsequent to that initial launch of Freestyle Libre. We introduced a series of digital health tools that allowed the user to, well, I should have described it. With the original Freestyle Libre, a user scanned the sensor with their reader. Something very similar. It uses NFC technology, just like the Apple Pay that you use purchasing things. So before, they would have to scan the sensor with their reader. We introduced some digital health tools that allowed the user to scan the sensor right with their smartphone and see their glucose readings right on their smartphone. And we also added some cloud-based data management software and eventually a caregiver app that allows the a caregiver to see the glucose readings in real time on their phone as well. And you can imagine the utility of that for a family of a child with type 1 diabetes to be able to see their readings in real time on the parent's phone as well. We've had professional versions of the device, the Freestyle Libre Pro. We introduced Bluetooth to the Libre sensor. And what that did was, that's the Freestyle Libre 2 system, and that allowed there to be alarms from the device, so it could tell you when your glucose readings cross high or low glucose thresholds and let you know. We introduced a version of Libre called the Libre Sense biosensor for sports in Europe in 2020. That's available in 8 countries in Europe today, and it's used by elite athletes to understand how their glucose values affect their performance and their fueling to achieve their best results. And then we have the Freestyle Libre 3 system, which I showed you as well. And then our most recent clearance is for use of the Freestyle Libre 2 and Freestyle Libre 3 systems for use in automated insulin delivery, like those closed-loop systems I showed you. So the Freestyle Libre products are right now used by more than 4.5 million people in 60 countries across the world. They're with reimbursement in more than 40 countries. And each of those generations of the Freestyle Libre system, what they did was achieve glucose monitoring without the need for regular finger sticks. They provide unique insights into people's glucose readings, and they're really designed with access and affordability in mind to be able to bring continuous glucose monitoring to broad populations. And then these devices sit within a digital ecosystem that I described. So we have apps for each generation of the sensor, the Freestyle Libre and Libre 2 and Libre 3 apps. We have a cloud-based data management system where patients and their healthcare providers can see their glucose readings essentially in real time as they're uploaded to the cloud. And then they can get insights from the reports. And then that caregiver app that I described. Those apps, we're also partnered with a broader digital ecosystem. We're partnered with people like coaching companies, Amato, Weight Watchers, Health to Sync in Japan, Levels 2, and also with an increasing ecosystem working with insulin manufacturers and insulin delivery, where we work with connected smart pens and connected insulin pumps. So companies like Novo Nordisk and Sanofi, Bigfoot, Insulet, Tandem, Ipsomedcam, Diab, and for all of these products, it's the glucose sensor that sits at the heart of their glucose monitoring and diabetes care experience. I have a lot of disclaimers. And then, of course, for people with diabetes, they need to make numerous decisions every day about how to manage their diabetes. And with Freestyle Libre technology, from RCTs and real-world evidence, we have a growing body of knowledge that shows that the use of these systems leads to positive health outcomes. So we know we see lower hospitalizations, we see lower HbA1c, we see increased time in target glucose range, and we see a reduced burden from glucose monitoring. So having diabetes has a tremendous cognitive burden, the weight of having to test and figure out your insulin dosages all the time. And for people using the Freestyle Libre system, we see a real reduction in that cognitive load of trying to manage their diabetes. And in fact, just this past year, the Freestyle Libre was awarded the Prix Galien as the best medical technology in the last 50 years, which kind of blows my mind to see that, but an amazing recognition for the team. It was awarded at the Museum of Natural History in New York last year. And of course, we're not done there. We've recently announced that we have in development a novel continuous glucose and ketone sensor. So one sensor, one in the back of the arm, that would measure not just your glucose values every minute, but also your ketone values every minute. And we think that a product like this has tremendous promise to redefine the care for people with diabetes at risk of developing diabetic ketoacidosis, for women during pregnancy, for pediatrics, for insulin pump users. We also think that this may usher in the ability to use SGLT2 inhibitors for people with type 1 diabetes, and they're not currently cleared for that. But those drugs, while offering all sorts of benefits, have risks of euglycemic diabetic ketoacidosis. So if you can have a sensor that alerts you to increased risk of ketoacidosis, rising ketones, still at normal glucose levels, we think this may open up the availability of these sensors for more people with type 1 diabetes as well. This is some early data that we've published, some feasibility data. There's actually 3 sensors that are nearly overlaid on top of each other. You probably can't make them out. Over 14 days, the red triangles are reference ketone measurements. In this study, they were people on a very low carbohydrate diet, so they were achieving ketosis, and that's the variation ketones we see. And we can see the three sensors show very good agreement between interstitial fluid ketone levels, that's what the sensor measures, and the blood ketone levels. So great proof of concept feasibility for that. And this is work that we hope to enter into pivotal studies quite soon, actually. I talked to you a little about that LibreSense glucose biosensor for sports. This is Eliud Kipchoge, the world's fastest marathon runner, and he uses the Freestyle LibreSense, he uses the LibreSense glucose biosensor for sport as part of his training regimen. And we recently announced the Lingo line of products in development. So these leverage the Freestyle Libre technology, but for health and wellness applications, so that people will be able to measure their glucose and ketones for health and wellness to understand how their diet and exercise affects their energy, their weight, their sleep, things of that nature. So while wearables today, we're all familiar, we saw a great example of watches and rings that live on the surface and provide us insight into sleep and steps, but it's really as biowearables like Freestyle Libre and now the Lingo line of products that are coming soon that give people unique insights into what's going on within their bodies. And we really believe that this is the future of health, of precision healthcare, of being able to understand and get the unique insights from what's going on inside your body with devices like these. So I appreciate all your time today, and thank you very much.
Maybe one quick question before you go. You spoke a bit about extending this to the other end of the risk spectrum, ketoacidosis, but what about broader populations? Your sports product speaks to that, with all the attention on weight loss and cardiometabolism in general. Is that in the plan?
I think there are broad applications. So for example, the keto diet is very popular these days, and a lot of people stop the keto diet because they don't know if they're actually achieving ketosis or elevated ketones. So they stop because they're not seeing a benefit. Well, if you have a sensor that tells you when you're in ketosis, it gives you the positive feedback to be able to maintain that diet and to see the potential positive benefits from it. And it's quite different from a diabetic ketoacidosis, from diabetic sensor where there we have alarms to let people know when they have elevated or high levels of ketones. For the Lingo product designed for people with on a ketogenic diet, it'll be a very different system.
Yeah, makes sense. Well, thank you very much.
Of course, thank you.
So our next panelist is Vijit Sabnis. He's CEO of Eigen Biosystems. Vijit, thank you.
Thanks for having me today. It's so special to be here back at Stanford. And today I'm going to talk about an emerging generation of real-time biosensors using aptamer-based molecular switches. I recognize a lot of you in the audience. I was back here in Tom So's lab not too long ago. And these are the co-founders, and I thought I'd just share a brief story here. Tom and I went to graduate school more than 20 years ago and reconnected just a few years ago to launch Eigen. We're about 9 months old, and in partnership with Professor So, Joe DeSimone, along with doctors Alex Yoshikawa and Pavan Kapoor, we hope to commercialize some of the technology out of Tom and Joe's lab. So we are really excited about building biosensors that sense within dermal interstitial fluid. And ISF, as many of you know, is the medium through which cells and tissue get nutrients as well as expel waste. And what's really exciting about the dermal interstitial fluid is that small molecules within the capillary bed can translocate very easily into the dermal ISF. And this makes ISF a great fluid for doing measurements. What's exciting about this is that the concentration levels of small molecules in blood track very closely with what is also in ISF. It's not just small molecules that are present in ISF. Nearly every protein in blood is also present in interstitial fluid, with the caveat that larger proteins are going to be at lower concentrations in blood. The other exciting aspect is that interstitial fluid is accessible with minimally invasive devices, very similar to what Marc just showed us. This has been very successful. Just to continue on what Marc said, the CGM is the first broadly deployed sensor in ISF. It's being used by millions of diabetics, and we're super excited that other small molecules like lactate and ketone bodies will soon be on the market. What is really emerging here is that the world is beginning to recognize the value of continuous data streams. And as engineers and scientists, all of us are going to really understand what this means. But these are movies of what's going on in our body. And when you look at a traditional assay, which gives you a single time point snapshot, that's like a picture. Movies provide so much more information compared to pictures. And so this really begs the question, can we have continuous sensing devices for other molecules? After all, there are thousands of other molecules in our body. What is the limitation of not being able to measure other molecules? So with the CGM, they utilize an enzymatic sensing technology. For example, here I have a picture of glucose oxidase, which allows the electrochemical transduction between glucose, and that catalyzes a reaction that produces hydrogen peroxide, which can be measured electronically using a small probe. The issue is that glucose oxidase is an amazing enzyme. It's been evolved in the human body as well as other organisms for thousands and thousands of years. It's very well understood. It's very stable. The issue here is that there aren't very many other enzymes that can be used to detect other molecules. Enzymes are not a generalizable solution. At Eigen, what we're trying to do is integrate aptamer-based biorecognition with state-of-the-art microarray patches. And we're really excited because the merging of these two technologies together has an opportunity to create a platform for measuring nearly any small molecule in dermal interstitial fluid. Another thing we're very excited about is to do so using multiplexing. We think measuring more than one molecule at once, being able to see that relationship between those molecules, that full movie is really exciting and is going to unlock lots and lots of insights that aren't available today. We're also looking at architecting a very low-cost platform so that this can be broadly deployed, not just within the medical system, but perhaps also for consumers. So at the heart of our technology are aptamer switches. The aptamer is the key component of the switch. The aptamers have been around for about 30 years. They're synthetic strands of DNA which can be engineered to bind to a target of interest, particularly small molecules and proteins. By adding some additional structure around these aptamers, we can turn these into switches such that when there is a binding event, there's a conformational change that can be read out either electrochemically or optically. And this is really a reversible switch. So this binding occurs and then that binding lets go so that we have the ability to measure real-time changes in the concentrations. And this is very different from a traditional assay where a biorecognition molecule, for example, such as an antibody, wants to grab onto a molecule, never let go, so that that binding stays there and a measurement can be made and you're done. But in contrast, these switches are binding and unbinding in real time, and we're able to detect a population of bound state switches and then track the concentration level. Professor So has been working on this for about 15 years. This is some real pioneering work that was published about a decade ago with a microfluidic chip in which electrochemical aptamer-based switches were embedded along the length of that. And some really amazing work was done here where human serum was extracted from a person while doxorubicin, which is a well-known chemotherapy drug, was flowed through that chip. And you can see here the ability to real-time sense changes in concentration in doxorubicin in the micromolar concentration range. This work was further extended to take blood from a live rat. It was an in vivo study looking at bolus injections of doxorubicin in the rat. And here what we see for the first time are real-time PK curves coming out of the device with doxorubicin. And then keeping the device exactly the same, but changing that aptamer switch from doxorubicin to a kanamycin switch, we could now measure kanamycin in real time. And here you can see again that classical PK curve. This time the clearance of the drug is slower than compared to doxorubicin. But again, the first time real-time PK was able to be done in an on-chip sensor. This work has been extended more recently. This is a paper published 2 years ago out of Tom's lab, where the focus was on miniaturization and the demonstration of spatially resolved pharmacokinetics. So in this work, a tiny probe with 3 individual sensor regions was inserted into a tumor. Doxorubicin was injected into a rat. And what you saw for the first time was different areas of that tumor getting that doxorubicin with different amplitudes and a different time lag. And this is the first time spatial real-time PK was ever demonstrated. This kind of information is absolutely super valuable because, as you know, tumors have a different type of microvascularization compared to unaffected parts of the system, as well as the microenvironment around tumors, which has different metabolisms, can affect how that drug is uptaken within that tumor. So this was the first time that you could actually understand how a drug spreads within a tumor and how efficacious it could be. At Eigen right now, we're focused on one of the principal challenges in attempting to commercialize aptamer switches for real-time sensing. Today, finding an aptamer and turning that into a switch is largely a bespoke process. It's done manually with a lot of intuition. All of my SoLab former lab mates here in the audience are deep experts in this. There's a lot of rational design and deep intuition that goes into making a good aptamer switch. This typically can take, inside academia, over a year to make a single switch. This certainly wasn't going to work in the confines of a startup company. So what we did is we borrowed the playbook from the pharma industry, where we took robotics and automation and took everything that has been learned within Tom's group and figured out a way to automate the entire process. So at this time today, we're about 50 times faster than what I think anyone else is able to do. So today we can crank through new aptamer switches in just a few weeks' time. With the data that's coming out of that new automation system, we're starting to invest in machine learning to bring us out of the world of simply rational design and starting to give real computational feedback and assistance in designing these aptamers and aptamer switches. And we feel like with some more investment in a software infrastructure on top, we're going to be able to bring this process down to a few days, again, massively parallel. This allows us to look at targets of interest in ISF and very, very quickly understand proof of concept. Is this going to work? Is this not going to work? And I think that allows our business opportunities to open up. As Marc said, there's lots and lots of different applications for continuous sensing. We're looking at consumer applications, management applications in mental health. We're looking at real-time organ function applications, for example, the kidney and the liver. We're also looking at, as I mentioned earlier, real-time PK and PD. We're really excited about this last piece. There are a number of narrow therapeutic index drugs where titrating that drug in at exactly the right level is much needed. Also for early stage pharmaceutical development, being able to measure a drug, understanding PK, could save a lot of time. In fact, we think by getting a sensor and deploying it very early on, we could save a pharma company at least a year in understanding how their drug is metabolized within a population. Now, what we've done with this aptamer engine or aptamer foundry, as I've been referring to, is our lab has only been up for a few months. We've taken a couple of shots to see what we could build very quickly. And here's some hot off the press data that I'm happy to share with you. Operating in the millimolar concentration range, we like to use glucose as the canonical benchmark to see if what we're doing is working. So over on the left-hand side is really our first shot at making a glucose aptamer switch. This really probably isn't something we're going to pursue commercially, given how successful the other companies in this space are. But it was a good first canonical example of can we do this really, really rapidly. And I'm happy to say we were able to do this in just a few weeks' time. Over on the right-hand side is real-time potassium sensing. Again, our first shot at this. The switch is not quite tuned to meet the time dynamics that we're looking for. But again, first shot, we're able to meet the, actually measure above and below the entire physiological range. Now, millimolar concentration levels are great, but there are so many molecules that operate orders of magnitude below that. So our next target was, could we get down into the nanomolar range, about 6 orders of magnitude lower than this? There are many drugs and many molecules that need to be measured in that range. This is an example of some data that just came out here in the last week. Our first shot at looking at real-time switching in a nanomolar concentration target. Here we were able to measure down to the nanomolar region. This is very exciting, particularly at the speed at which we're able to develop this switch. And now we're going to be pushing down even lower into seeing what the real limits of this technology are into the picomolar range. And again, by being able to span nanomolar, maybe picomolar, but nanomolar, all the way up to the millimolar range, we think the opportunities to really look at a lots and lots of different molecules is really big. So in conclusion, we're very excited about aptamer switches having a real opportunity to be a platform for next generation real-time biosensors. We think the real key here is to enable automation along with machine learning to really make this go quickly so that we have a chance to build a business. And with some success, we think there's going to be lots of opportunities around consumer, clinical, and pharma applications. With that, if any of you have any questions or ideas on applications, we're really open to collaborations and partnerships. Just drop me a line at info@eigen.bio. Thank you.
Maybe one question before you go, and we can discuss this more during the panel as well. I mean, the aptamer switch results are remarkable, and the work of the lab has always been quite impressive. But it seems to me an analogy to the continuous glucose monitoring. For any analyte, you'd have to demonstrate two things. One, that across the whole dynamic range that you want to measure,
Yep.
there's a actual equilibrium with the interstitial fluid.
That's right, yeah.
And then second, that you don't have, particularly at nanomolar and picomolar ranges, interfering small molecule species. So is there a way that that kind of biological confounding, which has, caused problems in other examples, that the aptamer technology solves it, or you can project it earlier in your design process?
So it's a great question, and it was too much detail to go into this talk, but these aptamer switches can be selected to be incredibly specific. So one of the things we're doing, and again, I didn't go into it in how we do the aptamer selection, we are trying to mimic the in vivo conditions in which we select the aptamers. Right. So when they work, and we have to be very smart about this, we have to include as many interfering molecules as we think so that we find the one or the few that can work. Otherwise, we would put all the risks downstream and probably we wouldn't be very successful. But that's the really powerful thing about this aptamer switch selection technology, which Tom has been working on for the last 15 years, is you're actually selecting the switches in the medium of interest. And if you think about antibodies, the way antibodies evolve, they evolve inside of our bodies where they are seeing all the interfering molecules, all the other stuff in the body. And so we're trying to take a piece of that. We can't mimic everything in the body, but we're trying to take a piece of that artificially and do that on the benchtop. And so that's really one of our powers in terms of moving forward with this.
Great, yeah, makes sense. Thank you. We'll take other questions during the panel. So I think our next speaker is Ashraf, Hanna, CEO of Genalyte.
Good afternoon. So I'm happy to be here and tell you a little bit about the technology we have at Genalyte. And I think the easiest way to share that is tell you a little bit about my son, who a while back asked me, "Dad, what kind of car did you drive in high school?" And I said, "I drove a Chevy Chevette." And he turns to his sister and goes, "That is so cool. Dad drove a Corvette in high school." And to his credit, they only differ by two words, two letters. But a Corvette and Chevette are really kind of different if you've never seen them. And other than the obvious kind of coolness factor, there is performance. There's an engine difference in the cars, and there's also a really important radio difference. My Chevette had a really great AM radio, and the Corvette had a fancy FM radio with quadraphonic. You may have not heard that term since the 80s. It's called surround sound now, but it had all these different features. And this is important because of the way immunoassays work today. And if you remember, by the way, FM is much better than AM. You get a lot more fidelity, a lot more capability. And the way immunoassays work is This may be very simple for some of you, but just want to bring folks together. The little antibodies are put on a surface, and they're targeted to a specific antigen that you're trying to find, and that when the antigen binds to it, you then send another antibody, and that antibody is called a detection antibody, and it's linked, it has something on it that allows you to see it. And what that allows you to see is that binding process, and it's typically something like light, radiation, or a color change. In fact, the sweat talk we just heard from Professor Rogers is just, you see a color change to see some underlying process. Very similar to actually what we do with radios. Radios are just a way of taking a small signal, like my voice, encoding it onto a carrier wave, sending that carrier wave miles away, and then decoding it. So this does the same thing. We get a small signal, and we encode it into, in this case, some measurement, and then we measure that thing. And this is basically an AM signal. You measure the quantity of light coming out just like an AM encoding process. What we have done at Genalyte is something, a really whole different platform. And it is, I would call kind of an FM signal. And what we do, this might be a little better pointer. Yeah, we make these little rings that are a couple microns in size. We put them on chips, and these rings have two features on them. One is a line, and one's a circle. And the line is just a fiber optic. It's a linear waveguide. And you send a signal down that waveguide, it comes out the other side like any fiber optic. Unless you happen to hit a frequency of light that resonates with the circle. In that case, nothing comes out. And what you see is an absorption peak. And it's not much different than spectroscopy, where you see absorption peaks, and that's due to electron levels. In this case, it's a man-made absorption peak that's set by the parameters of this chip. We then take that chip, and we put antibodies on the rings. And then when you run that, when you scan those frequencies again, you see an absorption peak. And in this case, it's this blue peak. When you run a sample over the chip, those antibodies bind. And now when you scan the frequencies, you get an absorption peak, and you see that difference has shifted. And so what you're doing is you're getting basically now a FM signal. You've got a frequency modulation due to the binding. Much, much higher fidelity, much higher signal to noise than you can. And anyone studied physics, or engineering, measuring frequency is something we can do really, really well. So we can see very little things here. In fact, we have charts where when you run the sample over here, if you plot the delta lambda here, the wavelength shift over time, you'll see that go up. And as that goes up, that relative shift, that's more and more antibodies binding until it peaks out where all the antibodies on your chip have bound their antigen. So you actually are getting real-time dynamic observation of the process of the antibodies binding their antigens. And because of that, we can actually do really, really cool immunoassays on a much smaller scale. And we can do a bunch of different ones. We can put down the antigen that you're looking for and look for IgG or IgM binding to it. So this is maybe autoantibody tests. We can do sandwich assays, which are the most common ones. This is TSH, HbA1c, typical laboratory tests. We can amplify those by adding a little bit of a carrier, not anything that transmits or sends a signal, but just has some mass, and that mass itself affects the signal. When we do this, we've gotten down, we're talking to about 10 picograms per milliliter. We've regularly gotten to 100 picograms per milliliter, but that's a really, really low level of capability of being able to see some of these antigens. And of course, competition assays, very common to like vitamin D assays in today's labs. One other feature we have built in to make this much more accessible is the ability to recycle. But before I do that, here's how we've actually made the chips. We've created chips, they're about 1 millimeter by 1 millimeter, 2 different flow channels, and each one has 16 sets of 4 different rings. And we put in antigens on all of these different rings. These are microfluidic channels that are separated, and that allows us to do either 2 patients at the same time or 1 patient with up to 32 antigens. So that's a big way of, that's how we've actually optimized that. The reason we do 4 is that this is still a young technology, and we measure everything 4 times and take the average in case there's anything that's off there. And so we can basically do 32 tests at the same time on this chip that's about 1 millimeter in size. One other feature is because these antibodies are covalently bonded to the rings, we can recycle them. We actually will run the test, take the measurements, and regenerate the chip by running a series of acid bases and other chemicals to have the antibody release their antigen and then restart again. The longest time we've been able to do that, we've been able to do 25 patients in a row on the same chip, and not only does that reduce costs, but some of these components and reagents are actually very difficult to get. So the more we can make that consistent and easy to use and reusable, the easier that is. So this is very interesting. It's actually pretty cool science. It's what made me come to this company. Where is it today? Two parts. First part is we've gotten about 36 different publications, which I think is an important part of being a small company, being transparent and sharing what's going on and having it be reusable by other people. All of these have been done at the specific institutions. The most recent one that I'm excited about is at the University of Michigan did an Ebola test. And this test was for a glycoprotein that's secreted as soon as you're infected with Ebola. They were able to show that they could get a signal within minutes of infection. The fastest test available. They got down to 1 nanogram per milliliter. And it was actually quite published in Cell. Others in Science, we've gotten cancer biomarkers, a panel of 12 cancer biomarkers, myositis panel. And of course, during the pandemic, we did a lot of SARS. We created a SARS chip, which was really reviewed quite significantly. In fact, we sent a machine to North Shore Hospital, to Mayo Clinic, and to Scripps. They all ran, this is a SARS antibody test, where you take a blood sample, and for us, it's only 20 microliters, which is about half a drop, so a finger stick worth of blood. And they would compare our machine to the other machines they had on site, and in all 3 locations, we were the most accurate, and it was because we were most sensitive. Mayo Clinic looked at that in particular, and looked at the time after infection when you could see antibodies, and we could pick it up almost a day before anyone else could see a signal. So it's actually very, very promising technology. And if you've seen an immunoassay machine, the smallest one's about the size of this podium. I would say the largest one are about put two or three of these together, and our machine's about the size of a desktop. So our goal is to be able to use that in more of a point of care setting, hence the topic we have here. So how do you do that in regulatory wise? Because one of the hurdles is some of these tests have to be reviewed by a certified lab scientist before they're released. So you don't just put your blood in a machine and it gives you a number, but a human being has to review that the machine was running at the right temperature, had the right reagents, you got enough blood in sample size, and releases that before a doctor or patient can use it. So one of the other things we built is what we call Cloud Lab. And this is a technology that allows us to wire all of these machines and let someone at a remote location see what's happening in all of these machines. And this allows us to do a few things. One is there's the constant monitoring to make sure it's running well. But second, we can run a control sample through it every day. We calibrate the machine every day. And as I mentioned, every sample that's run on there, the results actually come back to us, are reviewed by a lab scientist, and then released. So it actually acts as a central lab. And I joke around, we're like LabCorp, except instead of a big lab somewhere where everyone sends blood, our machines have really long cords, and they just happen to be at the site, but they're run by us. So this is actually a pretty important part of actually getting it out there without trying to ask the FDA, hey, change the regulations or make it a point of care test. So where do we go from here? So I would say, we have a product that could potentially be used at the point of care. It's much faster. And I think we have a couple phases for the business model. The first one is you just put it in a doctor's office. And if you go to doctors who are currently sending out their labs and to a third party and say, hey, for no cost to the practice, no change in what you're doing, we use the same tubes that you send to LabCorp or Quest, you just drop them in our machine and you'll get all your results while the patient's still there. That's a pretty easy sale. Folks sign up for that, and we have placed alpha test machines out there already. We've gotten feedback from that. We've just placed our first beta test machine in a veterans clinic, and that actually is revenue producing. And you start to see some of these results of just having real-time tests. We had a 79-year-old patient come in with back pain, and as he was being worked up, we did some tests, and he would have normally been sent home as he waited for the lab tests because it was non-specific back pain, and he turned out to be in renal failure. and he ended up going to the ER for dialysis. And if he would have gone home and waited 2 days for the lab tests, he would have most likely had, metabolic acidosis, or who knows what other complications would have happened. So the idea of making it convenient and catching real time has real medical benefits. The other part is there's a lot of issues with sending out blood. Approximately in the US today, about a quarter of patients don't actually go get their labs even when their doctor recommends they go get their labs. About 5%, much higher than I thought, of outside lab tests come back with an error. That's about 12 million times a year. Now, the doctor looks at that and says, okay, this can't make sense from the physical symptoms I'm seeing. This must be a lab error, so it doesn't always lead to bad outcomes. But that potential is there. So the idea of allowing real-time testing is really important. The other part is it allows you to do real-time testing for places that currently don't have any access. So the Indian Health Service or any remote town in the US, Lancet says about 50% of the world has no access to diagnostic testing. The number is closer to like 15% in the US, but all those places could now get a lab in a box, so to speak, and have that done right there. But I call that phase one, doing what we do today better. What is phase two? And I think phase two is taking a lot of the different tests that you see at major institutions, at Stanford, at Harvard, at Mass General. They have a great test that tells you your risk of an MI or a stroke within the next 12 months. There's a recently approved FDA test that tells you your severity of your concussion, or if you've had a concussion, and it's a blood test, and it correlates with CT scans. So you can imagine places where CTs aren't available to do a quick blood test that in 15 minutes can tell you if you've had a concussion. So this is what I call the app store model, that we take some of these tests today that are limited in scope and use and make them more broadly available. And this kind of brings me back to the Chevette. One of the things I had back then is in the trunk, you kept a lot of things. You kept oil. You kids won't remember this right now, but you keep oil, you keep radiator fluid, jumper cables, everything, because the car would break down and you got to be able to fix it. And there's no cell phones, and you had to know that. And our cars have gotten better, but what's really improved it is the sensors. And a good car today has almost 300 sensors in it, and they will tell you before you have a flat tire that your tire pressure is low, or you're starting to see the temperature increase, or just a check engine light. And all those things happen to catch it early. And I think this is exactly where we were going. When I was in med school, we were told not to do a test until you had a good differential diagnosis, or it's a hypothesis basically, of based on the chief complaint and the history and the physical exam. But as you've heard from other speakers, I think that's going to be backwards. As we make testing cheap and accurate and available, why not measure everything? And I think that's where we're really going, is that we're going to take all this model of tons of sensors that you have in car that lead to reliability, and this is actually our goal for our first chip, is to put all these on one or two chips and to be able to do all of that at the same time, and to bring that down to $100, or and to be able to therefore start checking things much earlier than you normally would have. And if you do that, that becomes a more of a standard, and you kind of pay for overall review. And this seems far-fetched, but it really isn't. We talked about Framingham, the heart study, or Project Baseline, and this is my Stanford connection. I think the dean who spoke is Project Baseline. I was actually on the other side at Verily getting the bills for Project Baseline and approving the budget for. It's like, I can't believe how much Stanford's spending. It's like, but it was really interesting time, and I think it's that kind of study that we'd need to do. We'd monitor 25, 30 different projects or analytes, and take a look at a longitudinal 10, 20-year study and use AI to help look out those signals and really help develop what I call a check engine light for humans. And of course, you can't end a story without a dad joke. So check in Genalyte. You can see our marketing message is getting prepared already. But thank you.
One quick question. We'll discuss studies and evidence generation, hopefully if we have time on the panel, but how dense can you make this chip, and have you thought about even perhaps running rudimentary sequencing reactions on the chip?
We can do sequencing, we can do aptamers. The limit ends up being the microfluidics, because for things like if you're looking for antibodies, so one of the interesting ones, both MSK and Alabama reached out to us, pediatrics, is a fever of unknown origin. And we can do like 64 in a little chip. And so you just put a bunch of different virus pieces and you see what antibodies you have and you can say, oh, you're infected with this. What ends up limiting it is the concentration of each of the molecules in the blood. You can't have something at a really high concentration being tested by something in a really low concentration because this one will flood while this one is still picking up the signal. So you have to separate the microfluidic channels into densities of molecules.
By the right dynamic range. Great. Please have a seat, and I'd ask the other panelists to come up as well, and we'll have a discussion. So great discussion. We'll have time for your questions as well towards the end. I mean, maybe one thing that we can start, we've discussed a lot of technologies, a lot of problems that have been solved, less about what is just over the horizon. So what problems are real today, and what really motivates you as to what is coming next, that is really realistic, not science fiction?
I'm happy to take that one. So for us in glucose monitoring at Abbott Diabetes Care, we do a very good job providing a glucose value to people so they're able to use that to make better informed decisions. But how can we take that glucose value and really, I talked about the cognitive burden of diabetes, but to really provide more context around that, give new insights, include things about exercise and diet and things like AID devices, automated insulin delivery, where you can take the patient, the person, the customer, as much of the burden away from them. So to me, it's really about seeing glucose in that broader ecosystem of all of the things that are important in managing someone's diabetes. I also think for us, we talked about these sensors have great potential outside of just the management of disease. I think there's tremendous opportunity in health and wellness space for glucose and ketones and lactate and a whole series of analytes potentially. So for us, I think we've shown a really powerful technology, and how can we expand it and to make it more insightful and more applications for people with diabetes, but even beyond diabetes as well.
So, but it sounds like you're quite conservative about the spectrum of analytes that you think would be coming next.
Well, our technology is an electrochemical enzymatic sensor, and we have we're probably the world's experts on that, and we've done a great job on that. We do see lots of opportunities to extend that technology, but that is the area of our core at this point.
Yeah, that makes sense. Car, I know your company is working on things that are very well validated, but fail to reach a large segment of your customer base. So what's next for you? Because there's almost everything in medicine that has that property right now.
Yeah, I think for us, we just sort of think about, our first step is really getting the cervical cancer screening out there. But then once you have that trusted relationship with somebody, how can we be a conduit for technology and innovation coming out to market faster? So for us, we would be looking to people like this panel to be moving things through the FDA. And as soon as they become evidence-based, or ideally on a guideline, we can make sure that a woman's choice and experience and preference is actually what's driving that decision, not necessarily what makes a hospital system more money or what somebody's more comfortable with. I think that a lot of times innovation really gets stagnated for good and bad reasons, but really we want to make sure innovation's getting out there faster.
Others?
I am excited about generative AI, and we talked a little bit about this at dinner yesterday, but when I get data from a physician, whether hopefully in the future it's an at-home device or maybe, for example, an Abbott device, it's oftentimes pretty difficult to go back and figure out what do these measurements mean? Do I need a consultation? Am I using Google? And having an intelligent bot to be able to interact with and tell me, what does that data mean? What does it mean for me personally? And this is part of my job, but also my personal interest. I listen to, for example, a lot of health podcasts where I think there's actually much better information being given there rather than my primary care physician. And I'll oftentimes remember, a year ago they talked about this. It's a whole bunch of work for me to go back and retrieve that, look at show notes. Do I still have a subscription to that podcast? But if we can have bots that are trained on good data, good information, oftentimes which is not within the purview or what your primary care physician cares about, that I see someone being able to build like yesterday. Like I sort of expect this to be built like anytime soon. That's really exciting to me. I'm not sure that it will be deployed on that kind of time scale, but in terms of what's right on the horizon, I look at the ability to interact with our data, hopefully increasingly with wearables, where we can really understand what our everyday lives are doing to our long-term health. That to me is where I think there's a huge opportunity for wearables to be even more valuable than they already have been, just because we're going to get that intelligent personalization and interaction with the consumer at home. And it's not just wearables, but just really any point of care or type of device. And I know, for example, when I was an investor, one of the issues with point of care devices was, well, I got this maybe it's at a clinic, for example, I've got this device, it's maybe not integrated in, and I've got to get someone to look at the data. But again, I think there's going to be an intelligence layer that's put on top of that makes everything very, very rapid. And I think it should be just around the horizon for something like that to happen.
It's interesting, maybe others will have a perspective on this too. What are the limits of that? Because right now, most of the examples that we can cite within healthcare, or even analogies that we would cite outside of healthcare, are cases of summarization, completion, searching through information. They're not bounded in the same way that we require healthcare decision support to be bounded, in that we have to understand causality, it has to be explainable, we have to make sure that there are no crazy errors to the systems. So what are the limits of that in the context of what your company is doing?
I mean, Ashraf, you probably have a good, yeah.
Well, I think that's early.
Yep.
I think we just don't know yet. But those things that you mentioned, like looking for patterns, picking out data, are actually really helpful. Those are, things that can help us be much more efficient as physicians and get much more timely information to patients, which I think reduces stress and reduces kind of follow-on. I think all those things are actually really helpful. But the actual kind of long-term value of this technology, I think, is still TBD. I want to also comment on your earlier point of like what we could do today. One of the things I'm most excited about, and I shared as kind of our first phase, is just let's do today's tests faster. And every physician knows what today's tests are. They order them, they go out, and they get them back in 3 days. Typical physician practice spends 2 to 3 hours returning test results a day. So you have yourself or someone in your office calling up patients and saying, hey, I got your cholesterol level, and we want to start you on Lipitor or come in for a liver function test. And you can not only get better care by getting them information right away, you can actually make the physicians and their whole practice that much more efficient. In fact, the AMA came out with a recommendation of asking patients or physicians to order tests before you come in to your actual physician meeting, so that when you have your meeting, you have the tests. Still requires 2 visits. You come in a week before to get your blood test, and then you meet your physician. But that whole trend of making the physician's time more valuable, I think, is both going to be AI and diagnostic testing, so that they have just a much more valuable interaction with the patient.
Yeah, there's an enormous leverage for diagnostic tests already, and we keep inventing new ones. As leverage, especially in oncology, is quite high.
I think we see in the glucose space already that by the time a patient comes to the physician's office, the physician can already be seeing what their glucose readings look like in their LibreView software. And you can imagine a time not far off where, a physician, when they see that report, will get AI suggestion around for patients who look like this on the current therapy, who have glucose patterns like this. Here are therapies that have worked. So I think that's really in the very close future.
Interesting. Kara, any comments about?
No, I was just sitting here thinking perhaps it has to do, will we see consumerization of this push? So it's like there's this idea that it has to be perfect if a doctor's doing it, right? Like we're all regulated, you can't mess around. But everyone's out there Googling anyway, right? And so are we going to find this kind of consumerization of the chat bots going in one direction, and then we have to come back and almost allow a lightening of regulations so that people are able to kind of be driven to the right locations that then leverage this technology properly to be able to give people what they need? I don't know, but I could see it happening. I was surprised by how many people were talking about ChatGPT. Not just in Silicon Valley.
And if there are any questions from the audience, please stand up and we'll work it in. Maybe the next topic we can discuss is evidence, and evidence that we have to generate for any of these things to be practical. That evidence ranges at the simplest from just analytical validation in the laboratory, or in the most complicated, some of the points that Vijit and Ashraf made are about either measuring new things or measuring things that aren't measured as frequently. So they're going to require a lot of data to prove that they're valid. How do each of you think about that spectrum of evidence generation?
I'm happy to start that one off. For glucose in diabetes management, it was relatively easy in that there was an understanding that for people with diabetes that measuring and understanding glucose values is important. So the initial for FDA clearances, it's all about accuracy of the device. But in terms of getting reimbursement and broad access to the product, it's about showing that the positive outcomes from the use of the device so that their health economics lines up. So you do randomized controlled trials in type 1 diabetes, in type 2 diabetes, and earlier in type 2 diabetes. And we've gotten increasingly broad access. And just in the last couple of months now, Medicare covers continuous glucose monitoring for folks on basal insulin, which was opens up this technology to a much broader area, to a much broader population of people with diabetes. So it really is, its accuracy is just, that's table stakes to getting in, but in order to get broad access, it really is the RCTs, and we're seeing an increased use of real-world evidence. We collect, information on, we collect de-identified data from users, and there are certain applications we're able to use that to support reimbursement in countries. So that's been very helpful as well, but you really do need the data for access.
Yeah, so multiple audiences and multiple levels of evidence ranging from regulatory to change of practice to reimbursement. And I think a lot of scientists working on these core technologies are not necessarily aware of that trajectory, which is why it's important to discuss. Other comments? Anyone?
Well, I think people should feel happy that the FDA does a good job of regulating testing. I think it's a good and appropriate bar. As we start to kind of go down that path, each test is run like 12,000 times. You run it, same sample 20 different days with different operators to make different times of day to make sure you get the same results. So that, if you've ever traveled outside the US in many areas, aren't necessarily sure, but you can be pretty sure that if a test is done in the US, it's probably, and people are following the standards, it's actually pretty good. So I think that's a good bar for us. It does two things. It keeps out Theranos, and it lets the others through. But the harder part is the new tests. When you see something and you start saying, hey, this looks like a good correlation, that takes decades. And the one I mentioned at Mass General Hospital was that they had a whole data set of a bunch of different tests and outcomes, which were MIs and strokes. And they looked and they found these 5 analytes that seemed to correlate, and that was like an AI-driven process. And then they did a prospective trial, which took a couple of years. It's because of the downside of the FDA, and it might be some better ways to do that, or some faster ways that we could potentially be able to measure things like that allow some of these diagnostics to not take 4 or 5 years to actually get out there.
Do you think that process is helpful or a hindrance? Because most of the calls for regulatory reform, in particular LDT reform, they would cause higher standards, not standards that are not necessarily lower, but more tuned to the kind of observational real-world evidence type validation that both of you implied or mentioned.
Yeah, no, okay, that's a good question. It's kind of tough. So LDT is a slightly lower standard than the FDA approval. If the FDA approves your test, you can sell it and anyone can run it. In simple terms, and LDT is you self-approve it, you have the data, and it's almost most of the FDA tests, but you're the only one who's allowed to run it. It's not bad if you follow the rules. But it's wishy-washy enough that you can get around that, and I think that's our first clinic is an LDT. We've prepared the FDA package, but it's still in process of being submitted, and I think that's it's okay. The truth, the way we did it is actually we did it with UCSF. They ran a trial and showed that it worked good enough, so we felt comfortable, but it leaves room for bad actors.
I think there's a couple of questions from the audience, at least one. Oh, one.
Yes,
There.
thank you all for excellent presentations. My question is directed to Ms. Egan. Very nice work. I think my patients really appreciate being able to self-test. However, we are seeing an increased number of advanced adenocarcinomas of the endocervix. They require a little more direction in getting the speculum exam and the test device higher up into the endocervical canal. And I'm concerned seeing a soft, we use a brush to get up higher, a steel brush to get up higher to get a good specimen there. And I'm concerned about a soft cotton tip not being able to sample that endocervical canal area. And I wonder if you have any information on your reliability and sensitivity in that particular area for the adenocarcinomas.
Yeah, so for us, we're going after first the primary HPV. So the leading guidelines have changed where you can do primary HPV and not cytology. And so for there, you can use vaginal or cervical cells. And so for exactly your question, so we've so far had success with pap smear, and we've had 100% concordance when the pap smear has actually been not been atypical. However, in our study, it's actually a lot more normal, a lot more normal pap smears. And so in order to go after the pap smear or Syntec 2, we would do Syntec, we would have to do a lot more studies with more.
I think that'd be important because that's what's killing our patients. In addition, you have the option now by the guidelines to do HPV high-risk testing instead of or in addition to cytology. Are you doing both or?
Yeah, so that's what I was saying. So the primary HPV is the new guideline that says you can just do an HPV, no cytology. There's only 2 on the market, Roche and BD. We run our study on both of them. For the cytology, we are collecting the data to show whether or not, but that would be an entirely different study with different endpoints and kind of more advanced.
Thank you very much.
Yeah.
It's an important question. There's maybe a more general version of this, and I think we have an odd perspective sometimes in an academic institution. There are all kinds of cancer tests that work very well, but which patients don't access. And often there are substitutes for those that reflex to those tests. Cologuard's product being an example. There are many others. You could argue that liquid biopsy will be an example for many screen cancers. So how do you think about the trade-off between doing what is best in a pristine sense and actually making this stuff available to a broad population?
So for us, right now, what we're doing is we're going after things that are already on the guideline. So we're not taking a new test through and then trying to petition the guideline. Once we're through, we're just a collection device. So we're showing that it's non-inferior to clinician collect on primary HPV. When you think about new tests, I mean, it's one of the things that we think about in terms of there's just such a long path to getting on guideline, and that's hard enough, but then how do you then get it out to market? Like, so for example, primary HPV, which has been around, and for American Cancer Society is the preferred screening instead of a co-test, instead of cytology, still only has 2.5% market share. Wow. It just, no one's changing. And so I think about us as kind of like, hey, once you're evidence-based, once you're on guideline, we're happy to try to get things out faster. Might there be a world where people are more proactive to say, I want to be helping to drive this innovation, be a part of these studies? I do think there's a way for us to welcome and broaden the demographic that's taking advantage, especially in preventative care studies, which almost anyone could be a part of, and they don't know they're out there.
And of course, it's the same pattern in diabetes and cardiovascular disease and dyslipidemias in particular. Another question.
I hope I say this correctly. My understanding is that the quantum AI program from DeepMind has decoded all of the proteins in the human body. And so the problem is, why have not some of the world's labs figured out how to cure anything with all that available? Isn't it true that they've decoded all the proteins in the human body, the folding?
Well, it's true that there are structures of proteins now, and that not just DeepMind, there's a broad academic community that's able to predict structure from sequence. But that is just the first step in a long path in drug discovery and drug development. So it's helpful, even though those structures are not always great, but it's insufficient to bring about cures in the way that you're suggesting. I don't know if others.
What about the cytokines in microRNA that transmit problems from one organ to the other? Okay.
I mean, there are companies that are even producing artificially designed cytokines to do what I think you're getting at. So that exists. None of them have reached the clinic yet. Other questions? Well, let's discuss a few more topics. One is...
There's a question.
Oh, there is. Okay, I didn't see. Yes.
Sharon Newman, radiology. I have a quick question, maybe super naive because I don't think about too much about FDA regulatory processes, but maybe I should. Is that as you're creating all of these companies, what are some processes that you think are hindering your company more than actually helping your product? And vice versa, what are things that you weren't expecting to help you that the FDA processes are actually improving your product in the long term?
Yeah, great question. And maybe as you respond, you can describe also what has happened during COVID and after COVID for some of the things that you're interested in, but please, anyone.
So we have a very close relationship with the FDA and work very closely. There's been continuous glucose monitoring was originally regulated as a PMA device, pre-market approval, which is sort of a very high bar in terms of medical devices. There was, with the introduction of a de novo filing, continuous glucose monitors moved into 510K clearance. They're regulated as 510K devices, which puts in a lower bar. The accuracy is still required, but there are things that, for instance, process validation, how you make it, those things are less under the purview of the FDA. I think they really do hear and listen to patient advocates, advocates like people like you in the room on the caregiver side as well. I think the FDA is sometimes a little slow to react to things, but they're well-intentioned, and they take that in, and they do, they learn about the risk profiles of devices. And devices like continuous glucose monitor was originally considered to be a high-risk device, and now with an understanding of how it's used within patient care, they have adapted to that. So it's an incredibly important relationship that we nurture. I work very closely with our head of regulatory, and we have a great relationship with the FDA, but also regulators across the globe. So in Canada and Japan and Australia and Europe, and those are parts of the job working in medical device or diagnostic industry that I probably didn't fully appreciate coming in, but it is a big part of what I do and what my team does, collaborating with regulatory and those agencies.
Yeah, for us, it's been great that they do have high standards, right? Because this is a screening. Then do you want to make sure that it actually is going to replace, if it can, and have the same sort of net benefit to society? One of the things that's helped us, it's not specific to the FDA, but was more policy change. So as these guidelines have changed, one of the things that helps is that the primary HPV assay is a PCR-based test, and so it's much easier to do an at-home screening. And so we have tailwinds coming with us with the Last Mile Initiative, which was the NCI and NIH saying, now that the guidelines have changed, let's help get self-collect and at-home through. And so when we have conversations with the FDA, I think otherwise it would be a little bit more, they might be like, who is this tiny company and what do they think they're doing? This isn't happening. And instead, whenever we have a conversation, they're like, hey, we've already put the pre-work in. We just haven't seen something yet that has met the mark.
I think on our end, being a very early stage company, one of the challenges has been we've got a lot of options on what ultimately our device could look like. And we've been advised, do not talk to the FDA until you've got stuff nailed down. And it would actually be very valuable, even at this stage, to say, hey, look, we've got these options, these indications. Are there some, don't go zones? Because we can think about a lot of those. But we're not going to do that. We've been advised by many people, have things locked down more, before you go to that pre-sub meeting. So I wish there was almost a brainstorming meeting where it's, this is who we are, we're going to be coming to the market, we're going to be coming to you guys, but what do you think? And I think we've been discouraged again from doing that, and that's holding us up a little bit.
Is it discouraged because they think that they'll fixate on who you were instead of who you've become?
You're anchoring.
Or?
You have a conversation and you're anchoring, and that's scary for a young company like us, where I think it's really slowing us down, where we could go in, talk about a bunch of different ideas, get them de-risked, get approval, I should say soft approval. Yeah, directionally this is a good place to go. Probably would help me raise money a little bit more easily than having that risk a little bit further down the line. So I wish that would change.
I'd agree with that. I'd say they're excellent if it's a product that you know has been approved and you can get it through, and you're comparing to something that's already out there. It starts to be fuzzy, and you got to be really careful if you're doing something new. For us, it was the recycling of the chips and reusing them on different patients, and they didn't know what to do with that when we brought up. It's like, are you going to contaminate the second patient, or and it's just, and you got to be ready and have that thought through before you bring up a new thing. Otherwise, they will get stuck into math or the biology and maybe go down a wrong path until you sort it out.
Complicated and nuanced answers. I mean, I think everyone obviously agrees that having an agency that is responsible for patient safety is very critical, but it is a government agency, and the way that you deal with it is highly tactical, and that's not always intellectually pure, but it's just the reality. So it's tough to answer simply. Do we have time for one more question, or we're out of time? Stop. Okay. Well, please join me in thanking the panelists. It was a great discussion.